Now we'll cover Chapter 11, or the endocrine system. And in order to introduce the system, we are going to talk about the different kinds of hormones that we have, how we produce them, and their different mechanisms of action before we move on to the individual. Now, first off, let's remember the difference between an exocrine and an endocrine gland. So an exocrine gland is a gland that has a duct where the gland will secrete whatever it's secreting. Let's say it's sweat, for example, and then that sweat will be released through the duct or exit through the duct out to the skin. It could be making different digestive enzymes maybe, so that duct will lead those digestive enzymes to the lumen of the stomach and so on. So the presence of a duct means that it's an exocrine gland. An endocrine gland, on the other hand, does not have a duct. It is a ductless gland. The endocrine gland makes hormones. Those are secreted directly into the interstitial fluid, or the fluid that surrounds the cell, and then into the bloodstream. Okay, these are the glands that we are going to be talking about in this chapter. We are discussing the endocrine glands and not exocrine glands. So what are the structures of the hormones? So again, all of these endocrine glands make chemicals known as hormones. Those hormones lie into one out of three major structural classes. Either they're derivatives of the tyrosine amino acid, and we call them amines. Those include your thyroid hormones, catecholamines, and dopamine. Or the chain will put 2 or 3 amino acids together, make a peptide chain, or make it into a bigger protein, so peptides and proteins. Or they're derivatives of cholesterol, and those are your steroid hormones. And those are the hormones, for example, that are synthesized and secreted by the adrenal cortex and by the gonads, whether ovaries and testes. And steroid hormones, remember that they are lipid soluble. So remember that one bit of information, because it's going to help us a lot in figuring out how are steroid hormones circulated in the bloodstream and how do they act when they reach their target cells, all because of the fact that they are lipid soluble, where your peptides and proteins are water soluble. So keep that in the back of your head somewhere. We'll come back to it later. Now, how is it that the cell is going to synthesize and secrete a peptide hormone? Well, peptide hormones are proteins, so whatever we talk, everything that we discussed in regards to protein synthesis will apply to peptide hormones. So the process of making a messenger RNA or transcription, and then translating that messenger RNA into a protein applies here. The protein hormone, though, is not secreted and released as a hormone from the get-go. First of all, when we synthesize, we are going to synthesize a prohormone. Okay, so this prohormone is inactive, and in the packaging system that happens in the Golgi apparatus, that prohormone is going to be split into a hormone plus the pro portion. So we will store and secrete the hormone as a hormone plus that pro fragment that came with it. So you can see here, this right here is a molecule of proinsulin. Again, proinsulin is inactive insulin. You'd have to split it into its different parts so that insulin is activated. And you can see here, this NH3 portion, that is your insulin part. The blue portion is known as a C peptide. And then this part with the COOH at the end, that is your pro fragment. And you can see here again, insulin is released as proinsulin with that pro fragment attached, plus your C peptide. This would have to be activated in order for it to act as your insulin hormone. C peptide is important though, because it gives us an insight as to the levels of insulin over a good period of time. So insulin doesn't remain in the bloodstream for a long period of time, but C-peptide does. So if you want to get an idea about the levels of insulin, say in the past 2 to 3 weeks, you can look at the levels of C-peptide in the bloodstream. C-peptide has no function when it comes to the regulation of blood glucose levels, but it is a good indication of the levels of insulin since they are related. Now for your cholesterol derivative hormones, you can see here the structure of the cholesterol molecule, and you can see all of these hormones that are derived of it. Cortisol, aldosterone, testosterone, and estradiol. Estradiol is just one of the estrogens. Testosterone, we have different kinds of testosterones. And then we've got cortisol and aldosterone. All 4 of those hormones are derivatives of cholesterol. So how does the cell know what to do with that molecule of cholesterol? Well, that depends on the enzyme package that is found within individual cells. So you'll see here is how, kind of a schematic representation of how steroid hormone synthesis occurs. So that cell will take in a cholesterol molecule, and depending on what kind of enzymes are found in that cell, that cholesterol is going to change or follow certain steps to change into its final steroid hormone. And then that hormone will diffuse into the bloodstream. But remember, again, it is lipid soluble, so it has to attach to a protein carrier in order for it to be carried in the bloodstream. So this right here is the suprarenal or the adrenal gland, and that is the gland that sits right on top of the kidney. And you can see it's made out of the adrenal medulla, which is the blue part in the middle, and that releases norepinephrine and epinephrine. And that brown, orange, yellowish kind of adrenal cortex. Okay, the adrenal cortex is made out of 3 different layers, from superficial to deep. We have the zona glomerulosa, and that makes aldosterone. Aldosterone is all about salt, especially potassium levels. So zona glomerulosa that makes aldosterone, you want to relate it to salt levels. And then the midzone, that is zona fasciculata, and that is going to make cortisol. And cortisol does change sugar levels, so you want to relate it to sugar. And then the zona reticularis zone, and that's the deepest of the 3, and that will make androgens. And androgen is a male sex hormone, so you want to kind of relate it to sex. So your zona glomerulosa makes aldosterone, zona fasciculata makes cortisol, and zona reticularis make androgens, which means in the adrenal cortex is going to make androgens whether that person is a male or female. And again, your medulla is going to make epinephrine and norepinephrine. So depending on which level the cholesterol molecule will go to, each of these zones has a different set of enzymes in order to produce a different hormone. So we'll take a look at kind of a representation of how that happens. So again, your cholesterol will be taken up by all of those different cells. If the cell is in the glomerulosa layer, we'll end up making aldosterone. Okay, that again, it would have a different set of enzymes. While if it's in the fasciculata layer, it'll go through a different route to end up making cortisol. But if it's in the reticularis layer, those reticularis cells would have their own set of enzymes and end up making androstenedione. What about the gonadal production of steroids? How do we make these steroid hormones in the gonads? Well, again, cholesterol in the testis, it has the same enzymes that ended up making the androstenedione, just like here, the androgens in the zona reticularis. But it has an additional enzyme in order to take it one more step and make testosterone. And that testosterone will be secreted by the testis. The ovaries have these aromatase enzymes that can change either the androstenedione or testosterone into these different estrogens. So estrone and estradiol are both different kinds of estrogens. So in the ovaries, it's all about the presence of aromatase enzyme. In the absence of that enzyme, the ovaries will not be able to produce estrogen. So again, aromatase enzyme is extremely important to be present in the ovaries in order to make estrogens. How are hormones transported in the bloodstream? Well, this goes back to that fact that I asked you to keep kind of in the back of your head. Well, that depends on the hormone. If the hormone is a protein, it can dissolve in water, meaning that it can circulate in the bloodstream. Okay, so we have no problems there. Okay, so your peptide and protein hormones can easily diffuse in the bloodstream. But what about the steroid hormones? You want to add to that thyroid hormones as well, because thyroid hormones cannot circulate freely in the bloodstream either. So your steroid hormones and your thyroid hormones, those cannot dissolve in water. They're lipid soluble. So you want to coat them or cover them with a protein. They have to be attached to a protein shuttle or a plasma protein. Now that plasma protein will circulate in the bloodstream attached to that hormone until it reaches its target cell. Some very small concentration of the steroid and thyroid hormones can dissolve in plasma, and those are going to be called your free hormones, and it's the free hormones really that are active hormones, while the hormones that are attached to plasma proteins are not active. They would have to leave the plasma proteins in order to be activated. So you can see here that that protein hormone complex can become a free hormone plus that binding protein, and the reaction is reversible. It can go in either way depending on the needs of the body. Now these are the categories of hormones. So we got peptides and catecholamines. Again, those are your norepinephrine and epinephrine. They are going to be transported freely in plasma because, again, they are water soluble. Since they're water soluble, then the receptors are going to be found on the plasma membrane, and their signaling will be through second messengers. Their rate of excretion and metabolism is fast. Okay, so is their action. They produce their effects fast, and their effects do not last for a very long time. While your steroid and thyroid hormones, those have to be circulated in the plasma attached to a protein. And because they are steroids or lipid soluble, their receptors are found inside of the cell. The mechanism of action, because they're intracellular receptors, would be through gene transcription. The rate of excretion and metabolisms are slow. The way they make an effect or a change on the cell is also slower than proteins and peptides, but their duration, their effect lasts for a longer time. So although the onset of action takes a little bit longer, but their effect on the cell remains for a longer period of time, again, because they are actually causing the gene to be transcribed. So they are producing a new protein, and that change is more permanent than the change that happens through these second messengers. What are the possible fates of a hormone after it's secreted by an endocrine cell? So all hormones are going to be circulated in a bloodstream. They can either end up being inactivated or excreted in the urine or feces, or they can become activated by metabolism and reach their target cells. Okay, in a target cell, they will bind to a receptor and then produce a cellular response. So how does the hormone know that it's at a target cell? So this isn't from your book. I kind of got it from another class. But that right here is your endocrine secreting cell. It'll make a hormone circulating in the bloodstream. And we have all different cells in the body. How does the hormone know that it reached its target cell? And that's by the presence of these receptors. So when it finds the receptor, it can attach to it and provide an action or lead to a change in that cell. So this is a very kind of primitive way because some of the receptors we mentioned are inside of the cell, but I just wanted you to understand that it's all about the receptor in order to identify the target cells. Now for hormone receptors, we kind of talked about this when we talked about receptors in general, that cells have the ability to either increase the number of receptors, and we call that upregulation, or decrease the number of the receptors, and we call that downregulation. Now, something new that we haven't talked about, and that is permissiveness. Permissiveness means that the action of one hormone permits or enhances the action of another hormone. Okay, this usually occurs due to the upregulation of the second hormone. So I'll explain how that works with an example. So epinephrine has an effect on the adipose tissues. Remember, epinephrine is part of the sympathetic system. So meaning that we need a lot of fuel, we need a lot of things to burn in order to produce energy. And one of the effects of epinephrine would be to break down the adipose tissue or fat cells into fatty acids. We can use those fatty acids to burn and produce ATP. But epinephrine alone cannot really do that. So you see here, if you plot time against how many fatty acids are released, if you look at that orange graph, epinephrine can do it on its own, but it's not doing a very good job. Thyroxine has the same effect on adipose tissue, but again, it's even a worse job than epinephrine. But together, you see here that there is that permissiveness where now, because we have thyroid hormone with epinephrine, thyroid hormone is going to permit or enhance the effect of epinephrine on the adipose cells. And you can see here that the amount of fatty acids that are being broken down, or amount of adipose tissue that is being broken down into fatty acid chains and released, is a lot higher when they work together. So that again is known as permissiveness, where the presence of thyroid hormone permits and enhances the action of epinephrine. Now, what are the pharmacological effects of hormones? Pharmacological effects are different than physiological effects. So physiological effects means those are the actions in our body when they are within normal range. But sometimes we have to give, or individuals are given hormones as treatment for medical purposes. And that would be known as your pharmacological effect, because you're giving them higher than normal concentrations or doses. You can also see pharmacological effects in cases of diseases. So if there's hypersecretion of that hormone, maybe due to a tumor of the gland or what have you. So again, your pharmacological effects would either be due to a disease or due to a drug intake of that hormone, higher levels of hormone. Now, how do we stimulate or inhibit the endocrine gland to secrete a hormone? How is it that hormones or endocrine cells know whether to release the hormone or not release the hormone? Okay, so the input that acts directly on your endocrine gland cells to either stimulate or inhibit hormone secretion. There are a lot of factors. We'll focus on 3 of them. So here you can see that endocrine cell, and it will be affected either by the levels of different ions or nutrients, levels of neurotransmitters, or the levels of other hormones. Okay, and we'll give examples for all of these as we go. Most of them are controlled by negative feedback. So let's give an example of an ion or nutrient that could affect your endocrine cell secretion. So for instance, an ion, for example, potassium. Okay, we talked about aldosterone levels and how aldosterone is really related to salt, especially potassium. So if a person has hyperkalemia or high levels of potassium in their bloodstream, that is going to stimulate the release of aldosterone. Aldosterone will dump potassium in urine, which in science we call it will increase the secretion of potassium. It will increase the tubular secretion of potassium in the kidneys, which again in English means it'll dump potassium in urine and bringing that potassium levels back down to normal. So you see here that the change in ion level, the change of the level of potassium, led to the stimulation or the increase in the production of aldosterone. The opposite occurs if there is hypokalemia. So in the case, if a patient has decreased level of potassium in their bloodstream, that will inhibit the release of aldosterone. Okay, for nutrients, the infamous example of that would be glucose and the production of insulin from the pancreas. So if a person, after a meal, the glucose levels go up in the bloodstream, there's hyperglycemia, and that will stimulate the pancreas to make insulin. In between meals or overnight, when we're not eating for longer periods of time, there is hypoglycemia, and that will inhibit the pancreas from releasing, from secreting insulin. So again, you can see how the levels of ions or nutrients can change the secretion of a hormone. And let's take a look here. This really talks about the ions or nutrients, and that giving you the example of glucose that we just went over. So in hyperglycemia or increased plasma glucose concentration, that is going to stimulate the insulin secreting cells to make insulin. Plasma insulin concentrations go up, and the insulin will reach its target cells and increase the glucose uptake by these cells. So the glucose goes from extracellular to the intracellular fluid, and that will decrease or bring back the plasma glucose levels back to normal. So again, by negative feedback. Now, pathways by which neurotransmitters influence hormone secretion. How does the nervous system affect the release of hormones? It can either be through the autonomic nervous system, where, remember the adrenal medulla, that is kind of a specialized kind of a ganglia, and instead of having a post-ganglionic neuron coming out of it, it will release a hormone known as epinephrine. So just to remember what we're talking about here, if you guys remember this image right here, in the sympathetic nervous system, we said the preganglionic neuron will go to the adrenal medulla, and it is a very specialized kind of ganglia where instead of having a post-ganglionic neuron, it is going to release the hormones into the bloodstream. It'll release epinephrine mainly into the bloodstream. So that is, going back here, what we're talking about. Other glands can also be controlled by the autonomic nervous system, either to stimulate or to inhibit those glands. And there's a connection between the hypothalamus and the pituitary, whether the anterior or the posterior pituitary glands. We'll talk about them where there are different connections. So the connection between the hypothalamus and the anterior pituitary would be that the hypothalamus releases hormones, and those hormones are either going to stimulate or inhibit the anterior pituitary, while the hypothalamus also makes some hormones, and those hormones are going to be stored in the posterior pituitary and then released from there. So the posterior pituitary is not a real gland. It is pretty much a storage for hormones that are produced within the hypothalamus. Now, a hormone that controls the secretion of another hormone is referred to as a tropic hormone. So talking about the hormones coming out of the hypothalamus that would stimulate the anterior pituitary, we call those tropic hormones. Now at this phase, I'm going to ask you to stop and pause, and then before moving on to our second portion.